In this video, we will discuss about the comparison of exons and introns of different species, as we have discussed that when a gene is uninterrupted, the map of its DNA corresponds with the map of its messenger RNA. When a gene possesses an intron, the map at each end of the gene corresponds to the map at each end of the message sequence. Within the gene, however, the maps diverge, because additional regions that are found in the gene are not represented in the mature messenger RNA. Each such region corresponds to an intron. In genomic DNA, there are two introns, each of which contains a series of restriction sites that are absent from the complementary DNA. The pattern of restriction sites in the exons is the same in both the complementary DNA and the gene. The finer comparison of the base sequences of a gene and its messenger RNA permits precise identification of introns. An intron usually has no open reading frame. An intact reading frame is created in a messenger RNA, sequenced by the removal of the introns from the primary transcript. Moving forward, the structures of eukaryotic genes show extensive variation. Some genes are uninterrupted, and their sequences are collinear with those of the corresponding messenger RNAs. Most multicellular eukaryotic genes are interrupted, but the introns vary enormously in both number and size. Genes encoding messenger RNA, transfer RNA, ribosomal RNA, and polypeptides all have introns. Introns also are found in mitochondrial genes of plants, fungi, protists, and one metazone, as well as in chloroplast genes. Genes with introns have been found in every class of eukaryotes, archaea, bacteria, and bacteriophages, although they are extremely rare in prokaryotic genomes. Some interrupted genes have only one or a few introns. Most of the variation in the lengths of different globin genes results from length variation in the second intron. Let's understand it using example of enzyme dehydrofolate reductase. Although intron lengths vary, the first intron is always fairly short, and the second is usually longer. Most of the variation in the lengths of different globin genes results from length variation in the second intron. The example of the gene for the enzyme dehydrofolate reductase, a somewhat larger gene in 3 mammal species, the mammalian enzyme dehydrofolate reductase gene is organized into 6 exons: 1, 2, 3, 4, 5, and 6. The gene itself is long because the introns are very long. In 3 mammal species, the exons are essentially the same, and the relative positions of the introns are unaltered. But the lengths of individual introns vary extensively, resulting in a variation in the length of the gene from 25 to 31 kilobases. So we can say mammalian genes for enzyme dehydrofolate reductase have the same relative organization of rather short exons and very long introns, but vary extensively in the lengths of introns. So we can say that the positions of introns are usually conserved. When homologous genes are compared between different organisms, The lengths of the corresponding introns can vary greatly. Introns usually do not encode proteins. Moving forward, alternative splicing generates the alpha and beta variants of troponin T. Troponin T gene of rat muscle contains 5 exons, W, X, alpha, beta, and Z, but only 4 are used to construct an individual messenger RNA. So, let's understand it. 3 exons, W, X, and Z, are included in all messenger RNAs. However, in one alternative splicing pattern, the alpha exon is included between X and Z, whereas in the other pattern it is replaced by the beta exon, the alpha, and beta forms of troponin T, therefore differ in the sequence of the amino acids between W and Z, depending on which of the alternative exons, alpha or beta, is used. Either one of the alpha and beta exons can be used in an individual messenger RNA, but both cannot be used in the same messenger RNA. So, the polypeptides produced by the 2 messenger RNAs contain sequences that overlap extensively, but are different within the alternatively spliced region. Moving forward, alternative splicing uses the same pre-messenger RNA to generate RNAs that have different combinations of exons. Alternative splicing can lead to the inclusion of an exon in some messenger RNAs, whereas it leaves it out of others. A single primary transcript can be spliced in either of 2 ways. In the first, which is more standard pathway, 2 introns are spliced out, and the 3 exons are joined together, and long protein will be produced. In the second pathway, 2 exons are joined together. The second exon is excluded, as if a single large intron is spliced out. This intron consists of intron 1, exon 2, and intron 2. In effect, exon 2 has been treated in this pathway as if it were part of a single intron. The pathways produce 2 polypeptides that are the same at their ends, but one has an additional sequence in the middle. Moving forward, the exon structure of globin genes corresponds to protein function, but leghemoglobin has an extra intron in the central domain. Both alpha and beta globin genes have 3 exons and 2 introns in conserved positions. The central exon represents the heme-binding domain of the globin chain. There is a single myoglobin gene in the human genome, and its structure is essentially the same as that of the globin genes. The conserved 3-exon structure therefore predates the common ancestor of the myoglobin and globin genes. Leghemoglobin genes contain 3 introns, the first and last of which are homologous to the 2 introns in the globin genes. This remarkable similarity suggests an exceedingly ancient origin for the interrupted structure of heme-binding proteins. The central intron of leghemoglobin separates 2 exons that together encode the sequence. Corresponding to the single central exon in globin, the functional heme-binding domain is split into 2 by an intron. So, the exon structure of globin genes corresponds to protein function. But leghemoglobin has an extra intron in the central domain. Well, hope you have a better understanding of the exons and introns. Thank you for learning. Join us again.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.